the genomic and it's joint work with Guillaume Achaz, Anton Krombach, and especially Julie Marin, who did most of the work. So the group that I've been leading for the last 8-9 years is called SMILE, for Stochastic Models for the Inference of Life Evolution. It's hosted in Collège de France, and one of the questions that we are interested in is the study of macroevolution and the inference of diversification from, let's say, the trace that evolutionary processes, macroevolutionary processes have left in biodiversity data. And specifically, the question that we ask is whether we can infer the processes that have generated biodiversity as we know it. I listed a couple of examples of the processes that we would like to be able to infer. So, for example, when and what are the drivers of adaptive radiations? Can we detect and understand or identify the different geographic modes of speciation? I'm sure you've learned about allopatric speciation versus parapatric, sympatric, peripatric, etc. When can we detect divergent adaptation linked to different selection regimes in different habitats? Can we detect mass extinctions and its drivers, species selection, and a phenomenon that has been recently documented, which is massively documented actually, which is the introgression from a distant lineage. The fact that species that can be quite distant can exchange genes, and it may be sometimes adaptive. And so the industry we've been involved in for let's say quite a number of years, like I think the last 10 years or 8 years, is what is called the phylogenetic approach to diversification. In this approach, the only data that you take into account is the species tree, okay, the time-calibrated species tree obtained from molecular data, but you just forget about the sequences, and you just retain the phylogeny, and the diversification process is assumed to have been generated by a birth-death process, where species are just particles that can split and die, and the rates at which it happens may depend on a number of variables like time, lineage, the current diversity, age of the species, or a trait like body mass, or I don't know, the presence, absence of wings, whatever. And so the idea is to compute the likelihood of the mere phylogeny under such a model and infer these dependencies. There have been numerous methods that have been proposed since the seminal work of Madison et al. And even I should have mentioned Nimei Harvey in '94, which are called BC, Quasi, GOC, High C, and that are meant for detecting the dependencies of these speciation extinction rates on these variables. It has been so popular in the last decade that there has been up to 4, I think, and maybe more reviews on this topic. But there's a but. The first but is that phylogenies are not big data, and so the signal is limited. In addition, there are statistical problems like false positive associations. For example, you can sometimes detect a dependence of the speciation rate upon the number of letters of the word identifying the species. There has been non-identifiability issues that have been emphasized by Luca and Penel in Nature last year. And so there's a need to search for additional signal, and this signal can be brought by fossil data, but also from genomes, and I will focus on this additional data. Also, the other but is that species are not particles. They do not split instantaneously into 2 new species. And so I think we need to also open the species box, which is sometimes called the magnifying glass on the speciation process. And the direction that we have proposed is to consider models that couple the diversification process and the evolution of sequences, molecular evolution, to infer macroevolutionary processes directly from genomic data, instead of waiting to first reconstruct the species tree and then take the species tree as the unique trace left by the diversification process. There is actually one model which is extremely widespread and that has been referred to several times during the conference, which is the multi-species coalescent, and that actually couples gene trees and species tree. So here you can see one of these ubiquitous pictures of a species tree containing gene trees. And so the idea is that here the species tree is given, so it's a top-down model. You start from the species tree, and the gene trees are confined into the species tree, with sometimes some exceptions, which is, for example, on the picture, the gene flow event, which is undergone by gene 3, the blue one. And so the idea is that gene trees, the model tells you that conditional on the species tree, which is sometimes the unknown actually that you want to infer, gene trees are conditionally independent coalescents constrained to reside within the species tree. Okay, so they are independent, conditional on the species tree. And so the disagreements between among gene trees is mainly explained, apart from these one-off events of gene flow, mainly explained by ILS, incomplete lineage sorting, which is just the randomness of coalescences inside the ancestral species. And so what might seem actually quite paradoxical, but seems to be widely accepted, at least as a tool, is that here gene trees are shaped by the species tree and not the other way around. And so if you think about this model, except for the gene flow events, all the signal and duplication and loss, when it is taken into account, I won't speak about it, but Tandy Warner has spoken about duplication and loss of gene lineages. Apart from these events, all the signal on the diversification process is contained in one summary tree. All right. So once actually the gene trees are only a nuisance here, and once you've used the gene trees to infer the species tree, the whole history, the whole information on the diversification process is contained in this end result. And so I think that we need models of gene trees. I think that gene trees contain more information than just randomness of coalescences and noise compared to the species tree. And so what I want to pledge for is bottom-up models of diversification and models of coupling diversification and molecular evolution, acknowledging that first speciation takes time. So speciation is not a one-off event that occurs instantaneously where one species splits into two. Speciation is a byproduct of genetic differentiation, at least as a first approximation. And so there's no hierarchy between gene trees and species trees, not actually the species tree that shapes the gene trees, but really the contrary. Gene flow is continuous. It's not something that happens at some specific points in time, but can occur all throughout the phylogeny, provided the two species are not too different. Gene flow is an important cause of gene tree disagreement, and ILS is not the only one. And if you think of this occurrence of gene flow, of ubiquitous occurrence of gene flow, then there will be no mother-daughter relationship between species. If you think in terms of the signal left by the genes, right? Because if you define an ancestral species as a species that has hosted the ancestral lineages to present-day genes, then these lineages, these gene lineages, can be hosted by different species, and so there might be different, like several ancestral species rather than just one in the multi-species paradigm. And so the questions that I would like to ask today is if we can show that actually we can devise such a model that would be parameter poor. So I don't want to have like too many parameters, but also I want this model to be plastic, to be adapted to maybe some more specific questions that evolutionary biologists might like to ask. And the second question is whether we can simulate easily this model and infer its parameters from a sample of gene trees without reconstructing a species tree. Okay, so it's a kind of species tree-free method, like Katja talked about on Tuesday. So the gene-based diversification model that I want to propose is, I think, the simplest one can think of. So here we have 4 parameters. Each species is made of several populations that can exchange genes. So here on the picture, you see, for example, in the bottom of the picture, a gene lineage that is exchanged between 2 species. Here time flows downwards. And you can see the populations here as the gray columns. Populations can replicate at rate beta to produce similarity, but each population is subject to genetic drift at rate gamma, and the populations accumulate genetic differences, so that's the mutations that are shown by black dots, to produce differences between populations. And what is not here specified is that when the differences between populations are too different, gene flow decreases until the populations cannot exchange genes and are considering as an emerging pair of new species. And so this model is mechanistic. It's based on how we picture neutral processes of diversification. It's out of equilibrium because there's a constant production of similarity and new populations, but also constant production of differences thanks to mutations. And species are naturally emerging from these differences. There's no explicit modeling of physical linkage as in the multi-species coalescent, and there's only 4 parameters, but it's very slow to simulate and of course to make inferences from. So what we've done is to propose a backward model of which can be seen as the retrospective view of this let's say global picture, but which is not exactly the mathematical dual, right? Not exactly the time reversal of this model. So here the difficulty is that you want gene lineages that are ancestral to one single genome to not spend too much time in different ancestral species. Otherwise, today, they would have been differentiated and they couldn't be found in the same species. And so to model that in the backward view, we assume that non-homologous lineages ancestral to the same genome are attracted together so that they cannot spend too much time in different populations. So here, there are 4 parameters. The rate A is the rate at which, here you can see on the picture, non-homologous attraction in the middle. Lineages that are ancestral to the same genome are recalled to the same population. And so when A is large, so this A tunes the timescale of gene flow. And then when A is large, most gene lineages will be concentrated in 1 or 2 ancestral species. So we stick to one species tree. B is homologous attraction. It's really how some lineages of the same color are attracted to each other and then coalesce. So when B is large, there will be a short wait between speciation so that B can be seen as tuning the speed of reproductive isolation. C for coalescence, as in the multi-species coalescence. So large C means no incomplete lineage sorting. And D for erosion, the fact that backwards in time, some gene lineages will tend to exit from the species they're hosted by and visit an otherwise empty species. And so this parameter tunes the number of populations exchanging genes. So from now on, I will assume that C is large, so ILS can be neglected. So the nice thing with this backward model, which is meant to be the time reversal picture of the global picture that I've shown before, but I've shown previously, but which is not mathematically rigorously the same, the time reversed picture. It's a bottom-up model. It's coalescent based, so it's fast to simulate. It's mathematically tractable because if you look at, for example, the past of one single genome, the genes in this genome will be eroded and pass in different species and will be recalled together. And there will be a stationary regime of this process. And actually, this process is the same process formally as the Moran model with resampling rate A and mutation rate D. So in particular, the number of ancestral species to N genes is exactly the same as the number of alleles in E1 sampling formula, which scales like log N. And so this is quite reassuring that the number of ancestral species will stay small as the size of the genome increases. It's also sampling consistent, meaning that if you look at the past of genomes with N+1 genes, and you take one gene from this genome, one gene at random, you take it away, and you look at the history, the past of these N genomes with N genes, it has exactly the same as the process defined in the beginning with just N genes. And it's plastic because it can incorporate time inhomogeneity and gene-specific rates, whatever you want to add in this model to make it more realistic or to detect more specific events or events specific to some genes. And so, yeah, here's a slide that I will not show. And we've also done some inference, but it's kind of preliminary, as you will see. The information that we have used is gene trees assumed to have been already inferred. So assume that we have like 10 or 20 or 100 gene trees. And we want to extract the information from the differences between these genes. And so we've used a distance between trees, the Billera-Holmes-Volkmann distance and the Kendall-Colline distance. And it's kind of what we've done is actually ABC for a multidimensional statistic, which is the distribution of pairwise distances between gene trees. And so we compare the compared distribution of pairwise distances predicted by the model via Kullback-Leibler divergence to the data, and we minimize this divergence in parameter space. And so by doing this, what we can do is not really infer the history, the configurations of gene lineages inside species, but we can infer the parameters. Yeah, it's just a slide to show that why we have chosen one of these two distances, the Kendall-Colline distance, instead of the BHV distance. So we've considered these two data sets, a Bayer data set and a Finch data set that have already been published, 6 species for each one, 14 loci and 7 loci respectively. And here's how the likelihood landscape behaves. So on each row is a data set. Each column is a method. So the left column is the MSC model with 2 parameters. And on the right column, you see our model, the GBD model with 2 parameters. And you see that the likelihood landscape is much better behaved. And so we found much more easily maximum likelihood estimator. And here's the reason why. So here you see on each row, again, the same data set. On each column, a same method. And you can see in gray, the distribution of pairwise distances between gene trees. Okay, that's the data. And in blue, you can see the simulation with the best fit parameters. And you can see that with the MSC simulations on the left column, you have gene trees that are much more different than in the data due to incomplete lineage sorting. And that's something that we didn't get. And so meaning that on the right column, meaning that what we detect is gene flow rather than ILS. So GBD is a 4-parameter neutral model combining diversification and molecular evolution. It couples gene trees without a species tree. It allows for pervasive gene flow. It's easy and quick to simulate. And it's plastic because it allows for extensions like co-evolving genes, time inhomogeneity, whatever you like. But of course, there's a lot of room for improvement. We would like to prove that there is a rigorous duality between the two models, or maybe another forward model. And we would like to infer not only the parameters of the model, but the history of the configuration of gene lineages. So I'd like to thank my collaborators, especially Julie Marin, who did all the work and who was paid by the LabEx Memo Life. And I would like to add that Guillaume Thomas is a PhD candidate under joint supervision of Céline Skornavaca and myself on extending the inference method. Thank you very much.
Sorry, I'm doing the clapping for everybody. Questions? Anybody? Please speak up. Okay, so I mean, you mentioned that the difficult, once you have the species tree, the gene trees are no longer needed, right? Did I get that right? That
you mean the contrary, right? So we use the gene trees, but we don't use the species tree. So we don't need here in this model. So here in the multi-species coalescence that I'm showing here, you infer the gene trees and then you infer the species tree from the gene trees, and then you can like throw away the gene trees. In our method, it's the contrary. We don't try to reconstruct any species tree. We only use the gene trees to reconstruct a much richer history of gene trees traveling between like possibly much more ancestral species than present day species.
So do you have a like rule of thumb or like some statistic you could calculate to see when it is in any sense useful to calculate the species tree? Or, I mean, is it ever, I mean, I guess it's useful because it makes things faster if there is such a well-defined species tree, but is there some, or is there another reason for it?
No, you're totally right. So one thing we could do is use our model and estimate, for example, the parameter, the D parameter, for example, and make like a cross-validation proposing like p-values for this D parameter, like a statistical threshold above which what we infer is not distinguishable from a model without gene flow.
Mm-hmm.
Yeah, totally. Yeah, you're right. Yeah, we should do that actually. Yeah.
Okay. Can I ask something really, really dumb and uninformed? So what exactly is the definition of species versus population? So I understand in this model, gene flow is flow of genetic information between different populations. So there are separate populations and there is some amount of gene flow between them.
Yep.
These are things that have zero gene flow between. I'm just trying to figure out what the definitions are.
Yeah. So in the backward model, it's a tricky question, but in the forward model, species are self-emerging from the picture. So what happens is that you have these populations that replicate, and let's say imagine that in the beginning they're all the sister populations from the ancestral populations are clonal to each other, and so when they're clonal to each other, they can exchange genes, and so you can assume that they are from the same species. And as genetic differentiation progressively occurs, the exchange of genes will decrease in rate, because what I didn't mention here is that the rate at which you can exchange genes between two populations is the fraction of genes that are clonal to each other. And after some point, you will see some populations that will disconnect from the others because the differences that have been accumulating with the others is too important for them to exchange genes. And so if you think of like two populations being part of the same species, if they can exchange genes, then this relation will self-emerge from the equilibrium between replication of populations and accumulation of genetic differences.
Okay, but somehow this is not an issue for you going backwards, that you have to define which things are the same species or not?
Yeah, you're right. So yes, so we cannot, since we are always assuming that at least for some time, lineages coming from the same genome are attracted together and so are recalled to the same species. What we can say is that the species that contain genes which share descending genomes at present day are from the same species. We can define them this way. But that's really a definition which is conditional on present day observations.
Okay, thank you.
It's due to the fact that divergence, lineages that are in the same genome today cannot have spent, like conditional of being in the same genome today, they cannot have spent too much time in different ancestor species in the past.
I would like to ask a more general question, probably.
Yeah.
So, maybe about the future of the application of the model. Mm-hmm. So you mentioned that the main motivation is the macroevolution. So how robust would it be in application to macroevolution with such things that are quite common, it seems, as parallel evolution or parallel processes in evolution, short-term, long-term, and you also mentioned mass extinction. So how would you detect these things? So why I'm asking this, because you work with genes, so you're taking like a bookkeeping things. So you try to reconstruct the genealogy, but genealogy is not always equated with evolution. It's so phenotypical evolution.
Yeah, you're totally right. So here, we've assumed that the genes we are keeping track of are neutrally evolving. Yeah, you're right. So yeah, if we had parallel evolution, we would have like we would infer gene trees that are not, as you said, like equating time, and so it's a good question. So yeah, actually, it's a question that you can ask even in population genetics. How do you? I mean, Alexandra is one of the specialists of this question. How you model a selection backwards in time, right? Yeah, but here that's not something we've considered.
And what about the mass extinctions? So how would you infer it from this kind of data?
Yeah, that's a good question. Yeah, I didn't think about it. I know that if you look at just the phylogeny, we have some papers on how to detect mass extinction from the phylogeny under a birth-death process. But taking into account additional signal from genes, that's a good question. Yeah, I don't know. Do you have an idea around your head? No. Okay. Yeah, good question. Thank you.
Claude, you had a question or no? No. Okay.
It was answered by already, so that's.
Okay, well, I think we're out of time, so thank you, Amaury, again, and let's move on to our next speaker. If anybody else has questions, you can catch Amaury and Gather.